Alrighty, guys. We're going to talk about something that should be familiar from your freshman year of high school, transcription, but we're going to add in some more details. First, let's go over what I hope you learned your freshman year, the central dogma of genetics. A dogma is like a super important tenet or idea for something. So, this is kind of a central idea in genetics. Information flows from DNA to RNA to protein. Going from DNA to RNA, that's called transcription, and as information flows from RNA to protein, that's called translation. Today, we are going to focus on transcription, and our next lesson will be over translation. But before we can do this, we talked a little bit about the structure of RNA before, but now let's talk in a little more detail, especially about some of the specific types of RNA. Just like any other molecule that we would talk about, the structure of RNA determines its function. And the structure of RNA is going to be determined by its sequence. So I mentioned briefly in a previous lesson that RNA can take on these complex three-dimensional shapes, and based on that, it can have different functions. So we're going to talk about the three major types of RNA, and I'm going to have some images on the next slide for us to look at. So messenger RNA or mRNA is the RNA we tend to talk about the most. It carries genetic information from DNA in the nucleus to the ribosome out in the cytoplasm. Once it is out in the cytoplasm, the ribosomes will use another type of RNA called tRNA or transfer RNA that has on one end a specific amino acid that matches with what's called an anticodon on the other end of the tRNA. This anticodon pairs with a specific codon of mRNA, and based on that, it will add a specific amino acid. We're going to go over this in more detail when we are talking about our next lesson on translation. So this is going to happen at the ribosome, and this is going to be used to make a protein sequence based on the sequence of the mRNA. Then the third and final type of RNA is ribosomal RNA or rRNA, and we really don't mention it after this point. The most you might see is an AP test question that mentions some recent experiment talking about ribosomal RNA, and they'll give you the background information you need there. All you need to know at this point is that ribosomal RNA is a part of ribosomes. It makes up part of the physical structure of a ribosome. So let's look at this in some diagrams. Messenger RNA is usually just detailed as a single strand that's kind of spread out. We don't tend to see it as any kind of complicated three-dimensional shape. And so we usually see messenger RNA as just kind of this linear line of RNA nucleotides. tRNA, I think it looks kind of like a T in a lot of representations. So you'll see at one end it's got a particular amino acid. At the other end, it's going to have these three nitrogen bases sticking out that are going to be highlighted. That is the anticodon that matches to a specific RNA codon. And then ribosomal RNA combined with protein makes up the physical structure of the ribosome. So if we look at transcription in more detail, we have one major enzyme that's involved. This is RNA polymerase, and it does all the major tasks of transcription. So it's going to use a single template strand from the DNA, and that is going to be used to form a template for the new mRNA strand that's being produced. Usually we talk about mRNA being produced, but really any type of RNA is produced in the same way. And then the nitrogen bases are going to base pair and complementary nitrogen bases will be added. Usually RNA polymerase is just going to unzip a tiny segment of the DNA at a time, whatever is actively being copied. So let's talk about the actual steps of transcription. So RNA will attach to a DNA molecule at the beginning of a gene. We'll talk more about what determines that attachment and how that's regulated when we get to another lesson on gene expression regulation of that. So what will happen is first, RNA polymerase attaches. It separates a small segment of DNA at a time to expose the nitrogen bases, and whatever the template strand is, so in this case, this is the template strand, and you see it's adding in the complementary nitrogen bases in the form of RNA nucleotides. It is going to produce this RNA in the 5' to 3' direction, which means it's actually reading the DNA in the opposite direction, 3' to 5'. So that is something to think about if you're looking at one of these diagrams on the AP test. And then at the end of the gene, you're going to have the RNA polymerase disconnect, and that's going to release what's called a pre-mRNA. Now, in prokaryotes, this process is much simpler. At this point, since there's no barrier separating the DNA from the cytoplasm, mRNA can be affected and read as soon as it is released from the RNA polymerase. So in the process of transcription, you can have concurrent translation. And because of this, your RNA is not modified before translation. That is not the case in eukaryotes. Eukaryotes tend to modify our transcripts from mRNA. So there's a pre-mRNA, that's what's freshly produced from the RNA polymerase. It's modified, and then it becomes a mature RNA. We call these post-transcriptional modifications. We're going to talk about 3 major ones. Polyadenylation is the addition of what's called a poly-A tail. It's at the 3' end of the mRNA transcript, so it's basically at the end of the gene sequence. And it is mostly just a repetition of a bunch of adenines. So you'll see here in this diagram, and it was hard to find a good diagram for this, but you'll see here all of these adenines, that is the poly-A tail. Now, what does the poly-A tail do? It makes the mRNA more stable. It helps with export from the nucleus. We tend to think of the nucleus and its pores maybe as very passive objects that stuff can pass in and out like an open doorway. But in reality, they're very complex structures that will very highly regulate what comes into and out of the nucleus. So polyadenylation and one of the other post-transcriptional modifications will help RNA get out of the nucleus. It helps to prevent degradation by nucleases that are in the cytoplasm, and it also helps with translation by ribosomes. Then we have a methyl G cap that's added at the 5' end of the transcript. So this you can think of as kind of being at the beginning of the transcript, and the polyadenylation happens at the end of the transcript. So let's say we have in this diagram your mRNA transcript. This is the 5' end. This is the 3' end, and this thing will be read in this direction. There is a modified guanine nucleotide that is going to be added at the 5' end. This is called a methyl G cap, and it's at the 5' end. The methyl G stands for methylguanosine, and it helps a lot with initiation of translation because it's at the very beginning of the transcript, and it also helps the ribosomes recognize the mRNA transcript as something to be translated. Now, the most complicated part of post-transcriptional modification involves exons and introns. So any given gene, genes can be some pretty complicated structures, and they actually contain sequences that are non-coding. We call the gene sequences, they're the parts of a gene that are going to end up in the mature mRNA and are usually going to end up resulting in the actual polypeptide sequence or the protein sequence. We call those exons. They are expressed regions. The other parts of a gene are called introns. Introns I think of as in-between regions, and they are not going to be found in the mature mRNA. They are excised or cut out, and that's going to become important in a moment. We'll talk about why in a moment. And the thing that does this excision is called the spliceosome. So we call the removal of these introns and then the kind of almost gluing together of the exons, we call that splicing. And so the spliceosome is a large complex made of RNA and proteins that's going to help splice out or excise the introns and join the exons together. Now, why is this important? Why would you have these sequences in your DNA and go through all the trouble and energy of making them if you're not going to actually use them to produce mature protein? It's because of something called alternative splicing. So when you have these various exons, let's say we're looking at this gene right here. It has 5 different exons. Each of those will code for a different part of a mature protein. So you can actually splice out different combinations of these exons along with the introns in between them. This is called alternative splicing, and this allows you to use one gene to produce multiple proteins. This is why, even though we only have 25,000 genes in our genome, you can actually have 40,000, 50,000 proteins produced by the human body. So if we look at this, if this particular gene ended up being spliced so that all 5 exons were in the mature mRNA, then what would happen is you would have all of these protein domains. So this looks like it's probably going to be some sort of receptor protein. And notice that you have all of these coiled sections. Those are going to be stuck inside the membrane. They're going to kind of thread in and out of it. You have what will probably be an intracellular domain. It might involve activity involving a second messenger. You have this extracellular domain, which might involve binding to a particular ligand. It depends on the particular protein. You can do alternate splices, though, where you remove, instead of having all 5 exons, you remove exon 3, and it's not located in the mature mRNA. Notice that when you took out exon 3, you took out this intracellular domain. So now you have modified the structure of this protein, which means the function of the protein is different as well. Notice in the final splice, you've removed instead exon 4, and without exon 4, you don't have this extracellular domain. So this extracellular domain that was present in the full-length protein is no longer there. So again, you change the function of the protein, and therefore you change the structure of the protein, and therefore you change the function of it as well. All right, that is all. At the end of this, you produce a mature mRNA, and very quickly, I'm going to sketch out a mature mRNA. So you have your RNA, you have your methyl G cap at the 5' end. I do not know why this is doing weird things. I am no clue what's going on. And then you have your poly A tail at the other end. And then you have your various exons. And this is the mature mRNA that's going to end up exiting the nucleus, going to the cytoplasm, and being translated by. All right, next lesson will be translation.